Okay, I'm happy to invite today our speaker, Dr. Ramaraja Ramasamy, the Professor of Chemical Materials and Biomedical Engineering and the Associate Dean for Academic Affairs in the College of Engineering at the University of Georgia. He received his PhD degree in Chemical Engineering from the University of South Carolina and held positions at the Pennsylvania State University and the Air Force Research Laboratory before joining University of Georgia, University of Georgia in 2010. He also previously served as the inaugural chair of the School of Chemical Materials and Biomedical Engineering. His research interests are in electrochemical engineering applied to biosensors, biomedical devices, and energy storage and conversion. Excuse me. And today he's going to talk to us about rapid diagnostic methods for bacterial pathogens.
Well, thank you very much, Dr. Gordon, and I would like to thank all of the most determined students to walk from wherever you are to this building. So I'm going to talk about some of our research activities in the area of developing rapid diagnostic methods, particularly for bacterial pathogens. And this talk is mostly focused on biosensors for bacterial pathogens and the ways to detect bacteria in primarily foodborne pathogens. Before I begin, I would like to just give an introduction about my institution, where I come from. Is the red one the laser pointer here? It is. Yes. Yeah. So if you watch football, you probably know where University of Georgia is. I know a little bit about that. But if you are not a football fan, so we are located about 80 miles east of Atlanta. University of Georgia is the flagship institution for the state of Georgia. We're not as big as you guys in terms of population, but we are a major R1 research institution, and we are also the oldest public university in the U.S. It was established in 1785, just a few years after independence. These are some of the metrics about the university. We're about $500 million in research expenditures. We go up and down on the rankings. For last year was 15. 2 years back we were, I think, 12. So we're also ranked #1 for bringing new products to the market. So we're very much focused on technology commercialization. And a lot of the vaccines, not the COVID, but a lot of the vaccines that are in everyday practice, some of them were developed at the university. So I am from the College of Engineering, which is the youngest college of engineering in a major R1 institution. We were only 9 years old. The college was formed in 2012. We started from about 19 faculty and 300 students, and we're now at 2,500 students and about 93 faculty. So we're still a small college. We're a rapidly growing college within the university. So the university has 17 colleges, or actually 18, in medical partnership with Augusta University, and we are the youngest college in the university. So my research in my lab, I direct a lab called Nano Electrochemistry Lab. So in my research lab, we have our lab is focused on application-driven research. Our research is application-focused. So we develop fundamental tools and materials and technologies based on electrochemical science and engineering to be used for. It's a little bit weird because you see the screen there, but you can't use the point where you have the turnaround. So really no purpose in looking at that screen. So our research is broadly can be categorized into 4 categories, primarily on electrochemical biosensors, where most of our research activities and projects are. We also have some interest in developing materials and characterization tools for energy storage systems and biological fuel cells and biological solar cells. If you're interested in knowing more about that, I can chat with you after the presentation. But the focus of my talk today would be on the development of electrochemical detection methods and platforms for chemical and biological analytes, particularly bacterial pathogens. So we start out by just explaining the need for rapid diagnostic tools. This slide needs no introduction because after COVID, everyone knows the importance of rapid diagnostics. If you were in February or March 2020, when there was no rapid tool, rapid test kit for COVID, you're at the mercy of some lab that does PCR, and you have to wait for several days to get your result. If at all, you can actually get your result. I mean, if at all, you can get the testing. So I tried to look at the numbers of how many people lost their lives or became severely sick just because of the lack of availability of rapid test kit for COVID. It was in the hundreds of thousands. So the importance of rapid diagnosis cannot be understated. And more than just disease detection, if you look at other things, best example would be something like testing for flu. If you want to go see your doctor to see if you have flu, now they have a kit that they can get an answer right there in the doctor's office, right? If you want to check your glucose levels in the blood, you can do it at home. So these things are really, the way that these things should work is, you should be able to, do your own diagnosis at home or at a facility where they will give you immediate results. You shouldn't be sending it to a lab and waiting for several days for the results to come in. The idea is, if you can't measure it by yourself, you really can't manage it. If you have to wait for 3 days to get your blood glucose levels for the morning that you took the test, then there's really no point. And so the importance of rapid diagnostic tools in a point of care setting cannot be understated. Now this applies not only to diseases, it applies to a variety of things that you want to monitor yourself, perspiration, exhaustion levels, glucose, cholesterol, or creatinine, and whatever it is that you want to measure just to manage your health. So that's where there are several tools that can be used in the development of point of care or point of service diagnostics, and we are interested in developing detection methods based on electrochemistry for a range of analytes. Analyte means the substance that you're trying to detect. Now we can, our capabilities extend from anywhere from small molecules, you can measure the small volatile organic molecules, all the way to large substances like bacterial cells. They are still small, but if you look at the scale of things that we could detect, they are like over five orders of magnitude difference. Sub-nanometers, sub-angstroms, all the way to bacterial cells, which are several microns in size. So we have a huge range of, wide range of detection capabilities, all based on electrochemical detection methods. So our diagnostic capabilities include detecting plant diseases based on just volatile profiling of air around the plant. We would be able to detect the onset of infection in plants. This is really helpful for agricultural crop disease detection. And we have capability to detect microRNAs, which are actually biomarkers for cancer, early stage cancer. We also have capability to detect NPK, nitrogen, phosphorus, and all kinds of inorganic contaminants in the soil, runoff water streams, and so on. They look very much like a pH meter, and they work very much like a pH meter. And we also have the capability to detect bacterial cells. This is where the last portion is where my focus is going to be, bacterial cell detection for this talk. So if you look at the existing methods for bacterial detection, if you want to just detect the presence of a pathogenic bacteria, for example, really the only available method that is you have to go the molecular route. You either send it to a lab for a PCR or ELISA-based techniques, immunoresponse. Usually takes a few hours to days for confirmation. I would say anywhere, the earliest you could get it is like 24 hours. Sometimes it would take longer than that, up to 72 hours, depending on the type of analyte that you're targeting. So it really goes through multiple steps, and then you have to take the sample. You have to, in most cases, you will enrich the sample so you can grow more of the bacteria to get a signal. and then you go through one of these routes. So this is where the biosensors have an advantage, because biosensors could potentially reduce the total test time, or the total the total test time from several days to a few hours. Now, the requirements of any testing method, not just biosensor, is you should have a sensitive, or it's not as sensitive, it's actually a detection limit. You should have a detection limit that's around 1,000 cells per ml, 1,000 polyforming units per ml. That's similar to what ELISA PCR can detect. And you should have specificity. There's a few things, a few terminologies here. Sensitivity this is actually not sensitivity, that is detection. Sensitivity refers to how sensitive your signal is with respect to the change in the concentration of your analyte. So that is sensitivity. Specificity is how reliably can you detect your target and not get false positives, false negatives with a non-target. And then rapidity of the test. Ideally you want to get a test within 5 to 10 minutes. That's exactly how glucose meters work at home. You put a droplet of blood and you get answer within 30 seconds. It should be portable if it needs to be used for point of care or point of use applications. And then you should have minimal sample processing. So these are some of the requirements of an ideal detection system. Now, the advantages biosensors bring to this to the table for this application is they actually can pretty much meet all of these requirements and then some. So specifically, biosensors can be both qualitative and quantitative. Qualitative is it's not really a biosensor, but I'll use the example of pregnancy test. It's going to tell you yes or no instead of telling how old the baby is. So that is a qualitative sensor. Whereas a glucose meter is a quantitative sensor. It's not going to say you have glucose present, it's say how much glucose you have present in your blood. So that's a quantitative sensor. So biosensors have that advantage. Instead of a test like lateral immunofluorescence or even PCR, which are capable of detecting low quantities, but it's mostly qualitative. And you can have biosensors that are quantitative. There's various different types of biosensors. The most popular one is the optical sensors because of the variety of optical methods that are available, anywhere from fluorescence microscopy to SPR, and all kinds of techniques are out there. So there's optical biosensors include a whole range of different methods for detection. There's piezoelectric that measures things based on vibrational change. Colorimetrics based on color change. An electrochemical, which is where our expertise is. Electrochemical measures change in the current or impedance or potential or voltage of the electrode as a result of a detection event. Now, if you look at the components of a biosensor, electrochemical biosensor in this case, there is a transducer, which is the electrode, which actually transfers the signal transfers the detection event into a signal. And your analyte is the target that you're trying to detect. And you have something called bioreceptor, or we call it recognition molecule or recognition element. And these are usually biomolecules such as antibody or a protein or an enzyme, DNA, aptamers, or viruses such as bacteriophage viruses. So these things provide, I mentioned about specificity, these things provide the necessary specificity. When you want to detect COVID, you want to detect COVID, and you don't want to detect some other virus. And the way that you can achieve that selectivity is because in a COVID test, you have an antigen or an antibody that can actually bind to that particular virus, and that provides the selective detection. So all these things can serve as recognition molecules because they would help us achieve the selectivity that we want in our detection. So these are the recognition elements, like I said. If you look at the simple case of a glucose biosensor, which is the commercialized example of an electrochemical biosensor, that's how it looks. And the way it works is you put your droplet of blood on this electrode. The electrode has a small region where it's coated with an enzyme called glucose oxidase. Glucose oxidase can take the glucose, convert it into glucanolactone. It's an oxidation reaction. As a result of the oxidation, electrons are released, and those electrons are picked up by the electrode and it's sent inside. There's a potentiostator power supply inside, so it's sent inside, and it measures the current, and based on number of electrons is the current, right? So it measures the number of electrons or the current, and based on the current in amperes, it can trace it back to the concentration of glucose, because the current is proportional to the amount of glucose oxidized on the surface. So that's how the meter gives you really based on the current that it measures. So this is how a biosensor works in principle. And every other biosensor, electrochemical biosensor, works on a similar principle. Instead of measuring current, it may change, it may measure a voltage or an impedance change, but the principle is the same. There is an electrochemical reaction happening at the surface, and the result of that, some signal is generated and the signal is transduced to a transductor to the electrode surface, which measures it. Okay. Now, our goal in this case, in the case of bacterial biosensors, we have used enzymes, we've used other things, but the work that I'm going to share with you is using bacteriophage viruses as the recognition elements for detecting bacterial cells. So bacteriophage are nothing but viruses. I think I have a detailed introduction of how they work in the next slide. Maybe I should have brought that slide earlier. But bacteriophage are basically viruses. Some people call it phage. I'm used to saying phage. I guess the virus doesn't care, doesn't get offended. So bacteriophage is a virus, just like any other virus. It gets inside the bacteria's body. It's highly selective. So one particular virus is specific to one class of microorganisms or a very specific microorganism. So it's only going to affect that. Goes inside, it multiplies its DNA, and it makes multiple copies. It just kills the cell and just goes on to infect other bacterial cells, just like the viral process. Now, what they have done is they have used some fluorescent tag on the viruses. So when they make multiple copies of themselves, then they will have a lot of copies of virus with this fluorescent tag, and they shine the light, and you'll get a fluorescent signal. Now you can say, okay, so if this virus is multiplying, that means the target bacteria is present in the sample. So that's how they measure this. And similar approaches have been used. Here is a colorimetric version of that, and there is also an electrochemical version of that, and there's a surface plasmon resonance change. So all of these things have been demonstrated in the past. But the key challenge with all of these things is they actually wait for the complete lytic cycle of a virus. What that means is the virus enters the bacteria, and then they make multiple copies, kills the bacteria, and then goes to the next cell to infect. So it depends on the lytic cycle of the bacteria, which is not a problem by itself, but if you're looking for a rapid diagnostics, you have to really wait for the virus to make multiple copies. That takes some time. If it's a really bad virus, it makes a copy within 30 minutes. Some viruses, take it slower. Typically, it's around 30 minutes to 2 hours for one complete lytic cycle. I'll explain what a lytic cycle is. This is the bacteria entering in, and then it penetrates inside, injects its DNA, and then it makes multiple copies and replicates, and then makes all the proteins necessary for its assembly, and then it assembles itself, kills the cell, comes outside. So this process takes anywhere from 30 minutes to 2 hours. Now, one lytic cycle is not enough. You have to wait for multiple lytic cycles to multiply the viruses if you want to get that fluorescent signal. Our goal was to achieve detection without waiting for the cell lysis. Our goal was to achieve the detection soon after the virus attaches to the bacteria, within like the first few minutes, within the first 5 minutes or so. So that way you have a huge advantage.
I'm just curious, for bacteria detection, is it important to do quantitative detection, or qualitative detection will be sufficient?
Yeah, well, it depends. For food safety application, I think quantitative detection is important. I'll explain why. Because certain, if you don't want to measure just one cell. If there is one cell in a big bag of food, is it a no? No, not really. But if it's one cell in a small piece of, little piece of meat, then maybe that's more concern than, bigger batch. So yes, it is important for some applications. Yes. So, so the advantages of using bacteriophage virus as a recognition element is, it is unlike enzymes there. Yeah. Do you have a question?
No.
Unlike enzymes, they are much more easier to handle. They're not very fragile. And they are highly selective to the bacteria. They're mostly harmless to humans. As a matter of fact, there are some bacteriophage viruses that they add as food additives to kill the pathogens, food pathogens. They're present universally, and they're actually much less expensive to make. Once you have a stock, and then you can just propagate them. It's not like viruses. It's not like enzymes where you need to purify, and it's more expensive. And the biggest advantage, perhaps, compared to like using an antibody, is they can distinguish between live and dead bacteria. Bacteriophage is not going to kill a cell that is already dead. Okay, so it is that perhaps is the biggest advantage of using the bacteriophage compared to any of the other things that I showed in the earlier slide. There are some challenges. The biggest challenge is, bacteriophage kills the bacteria by orienting itself by sitting on the cells. So which means its tail should stick to the bacterial cell wall and its head up. So unless the virus attacks the bacteria in this configuration, it's not going to be effective. So that's a challenge. If you want to use this, you have to really orient them in a certain way so they can do their attack. So that's where most of our work was. So if you want to put these viruses on the electrode surface, and if you want them to go and capture the bacteria, and you can see that this is perfectly aligned, that's going to capture the bacteria, none of the other things are. Okay, so the goal is to go from here to here. So this is a random immobilization. What we would like to have is an oriented immobilization, where all of the, not all, most of them are oriented in a proper way to maximize the capture of bacterial cells for an enhanced signal. So, besides the biosensor development, majority of our work was focused on developing this immobilization chemistry to attach the viruses onto the electrode. So I don't want to go into the steps one by one, but basically what we did was we took the electrode, we modified that with a forest of carbon nanotubes, and we have an in-house developed immobilization method that we use. In addition to that, we are actually using an electric field to sort of orient the virus to attach to the surface. The way that it has been done is the carbon nanotubes was modified with PEI, which is positively charged, and then the virus itself is negatively charged, but the head is negatively charged and the tail is positively charged. So when you try to apply an electric field and you have a positively charged carbon nanotubes, there is a tendency for the virus for the head to get aligned towards the positively charged PEI. So we're actually using a charge-based immobilization strategy here. So that way you'll have an attack. These are all the viruses. You'll have an attachment of the bacteria onto the electrode surface. So here's the methodology. So you take the carbon nanotubes, you modify that with PEI. In the process, you're actually taking the potential or the surface charge of the nanotubes from a more negative charge to a more positive charge. And then because the virus and the bacteria are negatively charged, they are attracted to the positively charged surface. You can see that once you functionalize that with the PEI, you can see that the carbon nanotubes are much more dispersive and it's more distributed in the solution there in its true nano form. And we did some ATR, FTIR measurements to make sure these functionalization has happened, and after that we also tried to put viruses on top of the surface to see if the viruses are not only attaching to the surface, but also retaining their activity, which means they're retaining their infectivity. You can see after putting it on the electrode, all the blue dots of the bacteria are present on the electrode, and you can see slowly they're beginning to die, and after about 2 hours, or actually 70 minutes, within 30 minutes, half of them are gone, and about 60 minutes, most of them are gone. So which means the virus retains its infectivity even after immobilization, which is important because naturally they will be infective only when it's in the solution mode. And here is another confirmation that you can see this is a control. There is no virus on the electrode. Here is a small piece of electrode that was just cut out from that test strip. And you can see that it's a small, I don't know if it's visible, there's a small clearing of the bacterial lawn around it, which means the virus is killing the surrounding bacteria there. And you can see here, 7 is the control where we don't see that clearing of the lawn, but all the other electrodes have viruses, and you can see there's a small clear of the bacterial lawn there. This is for MRSA pathogen, Staphylococcus aureus, and this is E. coli. 2 different examples. So again, SEM just to show that yes, you have bacterial attachment. This is before and this is after, and you can see that yes, bacteria gets attached to the surface. So we did electrochemical impedance experiments for all our biosensor characterization testing. So these are just regular electrodes. They look very similar to your glucose test strips. They have 3 electrodes working, counter and reference. Working electrode is the surface that was modified with the carbon nanotubes and then attached with the virus. The virus is actually present on the surface. And you do an electrochemical impedance test. Now, I don't want to get into the details of the experiment, but it's basically a test that measures the just applies a small alternating current or alternating voltage, AC voltage, and it measures the electrochemical reactions at the surface. And in the process, it can give us a lot of other information. And usually, this is an equivalent electrical circuit. I don't know if we have electrical engineers here, but this equivalent electrical circuit is a circuit that could be used for fitting the data, impedance data, to get some parameters. So this is a typical impedance response curve. We call it Nyquist plot. You can see when there is no bacteria present. I don't know. So I think it's this one. And then when you have bacteria present at different concentrations, you can see the change in impedance. When you actually take the magnitude of this semicircle, which actually is the charge transfer resistance here, that would be approximately equal to the diameter of the semicircle, then you can see that is plotted here versus the concentration of the bacteria. So for this is one of the very early cases that we did for E. coli. This is a non-pathogenic bacteria, by the way. We got a detection limit of about 1,000 CFU per ml, which is the same as PCR or ELISA. And we have a reliable range of detection from 10 power 3 to 10 power 7, and it's pretty linear. You can see it's 95% confidence interval. This is not the greatest. I'm going to show you more results that's going to be more interesting and more exciting. Here is, okay, apologize for the quality of the figure, but this is another pathogen called MRSA, methicillin-resistant Staphylococcus aureus. It's a very important pathogen because it's a pathogen that has high significance for clinical applications. Most patients get it through surgical infections, and it's a very important pathogen. And it's also resistant to antibiotics, so it's very important that this is being detected. So you can see similar set of experiments and the calibration curves. When you don't have the bacteriophage present, the change in the impedance is not high, but when you have the phage present, you can see there's a significant increase in the impedance, meaning that the electrode is trying to attract bacteria and to the surface, and it's able to detect with a much higher sensitivity. And the slope of this curve would give you the sensitivity, and of course the intercept would give you the limit of detection. So the limit of detection in this case, you can see, has gone down by 1 order of magnitude. Now we're about 100 cells per ml. Not only in laboratory samples, like a buffer sample, but actually in actual plasma samples, we got about the same limit of detection, 100 CFU per ml. And you have a reliable range. No one wants to care about infection or detecting something that is in high quantities. People want to detect something that is in lower quantities, right? If it's in higher quantity, if you have a food pathogen in your food, if you have a spoiled milk, you can smell it and tell. You don't need to have a sensor for that. We really are looking for something that can detect lower quantities. So here is the specificity study. So we call it selectivity. This is not the most interesting graphs, but I'll show this because it's the only graph I have for E. coli. This is just the virus with no bacteria present, and the K strain is sort of the non-target strain. You can see that doesn't change much with the curves in this region do not change much with the presence of bacteria. But when you put your actual target strain, that is the green one, you can see a clear difference. And I believe the K strain is the non-target strain. You don't see any spots. If you put the B strain, you can see these spots here and there. They're hard to see, but I can point out There's one here, there's one there, there's one. So it's the virus is clearing the bacteria. Another specificity test with MRSA. MRSA is another pathogen, like I mentioned, and here it's much more pronounced here. The no bacteria case versus the non-target pathogens such as Pseudomonas and E. coli. They're all sort of overlapping, but when you have your target pathogen, you can see a clear distinction in the impedance. You can also see it here. In the presence of MRSA, you can see a significant impedance change. And then in the absence of MRSA, there's nothing. And you can see with other bacteria, there's not much change between no bacteria and the bacteria case. So which means that the electrode is selective towards MRSA detection. There is the third pathogen. I don't have a lot of results because these are published. A similar set of results, you can see that when you have the phage and we actually put another blocker called BSA to reduce the number of exposed sites on the electrode. I don't want to get into that in too much detail, but what has been demonstrated here is very similar to the previous cases where you have the impedance change with the concentration. These are the selectivity studies. You can see when you don't have any phage on the surface, your target is Listeria, and the electrode actually doesn't measure Listeria significantly. It measures actually E. coli better. But when you have the phage, you can see it is selecting Listeria as its target. So it provides the selectivity when you have the phage. So that's the demonstration of these two graphs. Okay, so I've spoken a lot about impediment detection, which is good, but the real challenge is in any rapid diagnostic tests, the challenge is not the detection, the challenge is how do you prepare the sample for the detection. Okay? So the real bottleneck for rapid diagnostics is the cumbersome sample preparation methods. You can see all the green things here are the sample preparation. You can see, you start processing the sample in whatever various different ways, and there is maybe an enrichment step, and there may be a secondary enrichment, and other microbiological ones. So this could take easily, 3 to 4 days if it's not a very established method. And this is the bottleneck, because there is really you can have a test that can give you an answer. You can have a biosensor that can give you an answer in 5 minutes. But if you require a sample preparation that takes 24 hours, there is really no concept of rapid diagnostics there, right? And it should work very similar to your glucose meter. You put your blood, gives you a reading in 30 seconds. That's the direction. Now, it's challenging because you can have bacteria in your pizza, you can have bacteria in your lettuce, you can have it in your milk or ground beef. They're not easy to take sample and process it the same way. So that's the challenge. So these methods use enrichment of the bacteria, which means they're growing bacteria to increase their colonies. What we are interested in is just take the given amount of cells and just try to isolate and concentrate them instead of enriching them. Enriching is it's the step that takes a longer time. So let me show you the methods that we used. We used similar viruses because we want to only collect the target bacterial cells from our sample. We use these viruses. We actually took magnetic nanoparticles. They're not truly nano microparticles, and we modified functionalized their surface with these viruses. Now so let's say you have a sample containing bacterial cells. You introduce these virus modified magnetic particles into the sample. They're going to attach to those bacteria because the virus attaches to the bacteria. Now you can pull it with a magnet, and you're basically going from 1 ml to maybe 0.1 ml here. So that way you're concentrating the sample. So that way you can do multi-stage concentration steps. Then you can introduce into your electrode to achieve the detection. So that's exactly what we tried. Here you can see this is before magnetic separation, which means these and the bacteria are mixed together. After you pull them with the magnet, all the bacterial cells are at the bottom along with the magnetic particles, and you can remove the supernatant out. Now you have a concentrated solution of bacteria that you could input or feed into the sensor for your detection. And this can be done manually, or this could be done through a microfluidic device like this, where the magnets are actually channeling the particles. We've done these experiments with different particle sizes of the magnetic particles, and these are some results, you can see there's a huge variation in the capture efficiency. So it's not really dependent on the particle size when you're dealing with microparticles. With nanoparticles, we haven't done the experiment because it's hard to obtain nanoparticles of iron oxide commercially. But what I was trying to say is we were able to capture, anywhere from 40 to 50% of the bacteria at any time, which is better than none if you want to go through the enrichment route. Some SEM images showing that the bacteria is attached to the magnetic particles, 3 different sizes of magnetic particles, and here is the virus. This is actually a TEM image, and this shows only one virus, but you can also actually see there is tons of other things in the background that's not really captured here, that is on the surface. So far everything was done in liquid buffer samples. Now it's time to do some demonstration using real food samples. So here is milk and just an extract of ground beef, stirred in water. And you can see the capture efficiency for both of these for the magnetic particles. So we actually use 2 different methods, chemical and physical immobilization methods, to attach virus to the particles. And you can see, obviously, milk, you have a better capture efficiency, and for the ground beef, it's not, just because milk is a much more homogeneous sample than the other things. So, this is about 20 minutes compared to the, 18 to 72 hours needed for the culture method. And estimated cost is around $4 per sample. And the capture efficiency we got was anywhere from 90 to 40. It's not really high. Here in the culture method, it's not captured because it's enrichment, so you don't really worry about capture there because you're growing the cells. So just taking one step further, instead of doing manually, we developed a microfluidic platform where you can send the samples and you have a circular, hollow, concentric circle magnet that sits on top of this surface. And when the fluid goes inside, so you send the fluid in this pore, it gets distributed, and the fluid is your contaminated food sample, liquid food sample, and it gets distributed. When it goes through the serpentine channels, the magnet is going to pull all the bacteria from the sample, and then all the liquid escapes out. Then you send another lower volume of solution, wash away solution, then you get it out. So here you started with a 10 ml sample, and then you washed it away with 1 ml sample. That way you get a 10 times enhancement in the concentration of the sample. And in this spray, we were able to increase our separation or capture efficiency from 40 something percentage to up to 80%, which is much better than just doing it manually. So what I'm trying to show here is this is not the end of the world. When you continue to optimize it, you can capture and recover most of the cells. So here is a demonstration of selective isolation of Listeria here. And the magnetic particles, the challenge with the magnetic particles is they can physically attach to any bacterial surface or any sort of bilipid membrane. So you end up getting all the non-target bacteria to some extent anyways. But our results showed that you have a preference to Listeria if you have the Listeria specific virus. So it does capture most of our target analyte, and then a little bit of some of the other organisms as well. And this requires further optimization, and just block out the sites so non-specific adsorption happens. So after we separate the samples with the help of a magnet, instead of introducing into the biosensor just by a regular gravity feed, we also wanted to aid the immobilization with the help of a magnet, not the immobilization, the attachment with the help of a magnet, because their bacteria are already magnetized, so to speak. So when you put it with the magnet, you can see the impedance change. This is without the magnet. But when you put the magnet, it's a significant increase in the impedance change. It's actually better because you get a better signal. When you get a better signal, you can get a higher sensitivity for your measurements. Higher sensitivity means you're able to tell the distinguish between 2 different concentrations very easily. You can see that with the help of a magnet, you're able to achieve higher signal for the charge transfer resistance. Rushing through, I don't know if I have time. Okay. So anyway, for just as an application in a foodborne pathogens, and I mentioned Listeria, I mentioned E. coli. These are some of the foodborne pathogens. And they have their economic impact is huge. Norovirus is a virus actually. Listeria and Salmonella, these are like most common pathogens. We were specifically interested in Listeria recently because Listeria is a deadly pathogen. Not as deadly as COVID, but compared to all the foodborne pathogens, Listeria is really deadly because it causes a high rate of hospitalizations and high mortality rate compared to other foodborne pathogens. The biggest challenge with Listeria is it can grow at refrigeration temperatures. And it can be present in solids, it can be present in liquid. It is one of the most difficult pathogens to deal with. If you're infected with Listeria, you're almost guaranteed to get seriously sick from it. Not like everyone would end up in a hospital and die, but it will definitely give you a serious sickness compared to a mild foodborne pathogen. So even though Salmonella causes more illnesses, Listeria is something with a higher mortality rate. So it's very important. And some of the things that I showed here for Listeria, the reason we picked the virus-based detection method for Listeria is if you wanted to use the same virus-based detection method for Salmonella, it's not going to be suitable because Salmonella has about 2,300 serotypes and all of them are pathogenic, and you can't have a virus for every single type of Salmonella, pathogenic Salmonella. Fortunately for Listeria, there's only one particular species of strain that is pathogenic, and we have a virus that can selectively detect that. But so it works much towards our favor when you use the virus-based methods for Listeria compared to E. coli. In the case of E. coli, yes, we do have some strains that can detect the pathogenic E. coli, but it will also detect non-pathogenic E. coli because that virus is not selective only to the pathogenic strain. So in that case, you may end up getting a positive signal that is really not coming from pathogenic E. coli. So you may get a false positive, which you want to avoid. So that's why Listeria is our focus for now, and of course MRSA is another pathogen that's serious. And the ongoing work is on Campylobacter, which is Campylobacter jejuni, which is another important pathogen. So what do you do from this kind of work? So you can potentially convert it into a point of care or point of service based device. You can develop something that looks very similar to a glucose meter to look for bacterial pathogens yourself. But just beyond bacterial pathogens, electrochemical biosensors have applications in a variety of different things. Anything that can be oxidized or reduced on an electrode surface can potentially be detected if we find the right kind of bioreceptor and recognition molecule for that. You can easily develop things like the things that you go to the doctor's office to get your annual physical exam. You get most of these things checked out anyway. Creatinine, glucose, cholesterol, all those things. We could develop a biosensor for each of those components because every one of them are redox active and they could be either oxidized or reduced on the surface and selectively oxidized or reduced. So you could potentially develop a personal diagnostic tool for especially for remote setting like this, where they don't have access to labs or doctor or healthcare facilities. And there's a huge application. I mentioned in the one of the earlier slides that the rapid diagnostic market, point of care testing market, is about $6 billion in the US, and it's going to reach $26 billion throughout the world in the next few years. It's currently at 18 to 19, so it's going to reach $26 billion by 2026, I think. So it's a huge market for that, and that's where we are interested in, taking our devices to the next step, taking our methods into developing devices for these applications. So hopefully I gave a good summary about electrochemical biosensors being applicable to rapid diagnostic testing. And the key thing here is the construction of the biosensor interface. What goes on the surface for the for supporting the biomolecule, what kind of cross-linking chemistry can we use to attach the specific bioreceptor, which is called the recognition molecule, whether it's enzymes or viruses. And really, if you look at it, it's analytical chemistry, and there is nanomaterials, there is engineering in there, microfluidics in there. So it's actually this kind of research cuts across multiple disciplines. In our lab, and there's some genetic engineering, synthetic biology there, because sometimes we have to modify the virus surface by genetically modifying we put a tag on the virus surface to help in the attachment. So those kind of things require some synthetic biology expertise too. So it actually cuts across multiple disciplines if you're involved in this kind of research. So I believe advancements in nanotechnology, material science, and even artificial intelligence, machine learning that that reads the sensor and sort of learns how the sensor works and help us optimize these sensors would help push the boundaries of electrochemical sensing technology. So that's my last slide. Hope I'm within time.
Yep.
Yes. Very good. Thank you.
So my question is about the specificity.
Yeah.
Not only because you can use this for the food industry, but also for clinical applications.
Yeah, MRSA is actually meant for clinical. Yeah.
And have you thought of on combining that technique? Because you were able to isolate with the phages the bacteria of interest, but because bacteria is very there are many bacteria, different. To combine that technique with, for example, more specific like PCR or polymerase spiral reaction, which is something that you could do without machines, and it could be like something.
We could. It really depends, because our one thing to remember is our biosensors take 5 minutes to detect. So once you introduce the sample, it takes, I say 5 minutes because we run the experiment in that way. You could actually do a rapid measurement and get result within 60 seconds. So when you're talking about the isolation step, right? So the isolation step where you collect the bacterial cells from your sample in a more effective way. So if you start with 200 milliliters of milk and there's only 30 bacterial cells inside, I cannot introduce all the 300 because now my detection limit is not sufficient enough. I want to just concentrate and get the cells out in small 100 microliters. Now my biosensor is sufficient enough for making the detection, and the detection limits are sufficient enough to detect those 100 cells. So this entire process can be done, let's say, if you use the microfluidic device, it takes about another 15 minutes or so to carry out this. Once you go past that half-an-hour mark, the virus is going to kill the bacteria because it is attaching to the bacteria. So you really have to do something that can be done within the first 30 minutes, or first 20 minutes, I would say. If you want to use the PCR, by the time PCR uses the sample, the virus could have killed all the bacteria.
Yeah, but the DNA is there. For example, the fact that you will not be able to find all the phages you want to select for specific.
That's true.
That's what I'm talking about.
Yes.
Because you can find.
Instead of using phage, you could actually put an antibody on the magnetic particle.
Yeah, but that would be more costly. I'm just trying to think. That's the main problem in clinical settings. Like when they do surgery, you can have bacteria in your sample, like in your patient, but you want to detect that prior.
Correct.
So you don't have to have the patient 6 months later coming to the orthopedic surgery and say,
right.
"Oh, I have this bacteria," and you would like to detect that in advance.
Yes.
But then the problem is that. So when you take a sample, how you can isolate from the sample a swab or a piece of tissue, like a biopsy, and then the specificity. You want to know which bacteria is, so you can treat the patient.
Yeah.
So, but that's why I'm asking that, because you can select from a group of bacteria, and then after that you can say, okay, this patient has this, this E. coli that could range from this tissue to this one. But then I want to detect the specific E. coli, and then you can do you can combine that with PCR. It requires a little bit more time, but at least you know. You can rule out some,
You can. I
some
mean,
part of the diagnosis.
I think I get your point. You can. It really depends on what you want to do. If you're okay with, you want to target one bacteria, but you don't have the specific virus for that, but you have a virus that can target a group of bacteria in that category or in that genus. Yes, I think it should simplify the sample treatment procedure to some extent for sure. What I'm trying to convey to you is, if the bacterial cells dying is not an issue, then it's this can be used for food application. For example, I want to know if my salmonella is live or not. If the salmonella is already dead in my milk, I don't care. So that's why it's important to see whether it's live or dead. And the importance of using viruses is because it can capture only live cells, not the dead cells.
That's one of the important things, because I can see that you can say, okay, this is live bacteria here of this type.
Yeah.
But then I want to know if this is the pathogen one. So after you have your sample, you can process that very easily to do even a point of care, which is not PCR, maybe PSR, which is another kind of enzyme.
And I still like to use PCR if PCR can accept those variety of samples that we prepare. That can be used for PCR. PCR is a reliable technique. There's no doubt about that. It just doesn't provide a way to detect something really in a rapid fashion. And otherwise, but it's a reliable technique. In any case, biosensors that we're talking about now are only a first level of detection. There's always a secondary confirmation for which we go to reliable existing methods. Just like you're always being sent to a specialist, even though your primary care physician knows exactly what problem you have. They send you for somebody who's authorized to say so. So that's exactly how I see this. This is seen as a first level type of detection. For clinical applications, instead of just bacterial pathogens, we can also develop sensors that just uses enzymes to look for creatinine, for example, or adrenaline, or epinephrine, or all those things. As a matter of fact, electrochemical sensors are being explored for heavily in the Army for they're called perspiration sensors. They measure a whole range of metabolic compounds in the sweat to look at the soldiers' exhaustion levels. So there's a variety of possibilities with electrochemical sensing. Remember, I only specifically focused on bacterial detection, but it can be extended to others.
Yeah, that's why I came here, because it was bacterial detection. I'm working on that, so I thought, this is very interesting.
Yeah. And the capture efficiency, the capture efficiencies I've showed here is around 40%. And even one of the slides I showed, even though it selectively detects Listeria, it also detects these other things. So that's the challenge. How do you minimize that? Is because when you have these magnetic particles, you have the virus, and if the magnetic particle is somewhat exposed, then any bilipid membrane can attach to it. Because bilipid membranes can form FEOP bond with any magnetic particle. In that case, you're not only attaching your target bacteria, you're also attaching other bacteria non-specifically. The way to minimize that is cover the surface, block off all the entry points. And that's a challenge when you're dealing with really small particles and a huge virus on top of it. You're not completely covering the surface. I'm sorry that I've been holding you for long.
No problem. Thank you so much for it was a great talk. I have a question about one of your slides. So you show that you compare milk with beef.
Yeah, ground beef.
Yep. And then in the case of like the ground beef, so the specificity technically significantly reduced in comparison to the milk.
I don't know if it's specificity. It's the capture efficiency, maybe.
Okay. Does it mean that when the sample is more complicated and enriched with different type of proteins, the specificity reduced?
Yes. Very good question. So the more homogeneous the sample is, the easier for us to use it. I mean, remember, your goal is to capture bacterial cells from whatever. And if that whatever is as smooth and homogeneous as possible, then your job is simple. All you have to do is just mix it with it, and then you get that. But if you're talking about a ground beef sample where you have half of that is particulates, like fat particles, protein globules, and all that, then it sort of messes up. So we actually, I haven't shown here, what we actually designed a microfluidic platform where we had a multi-stage filtration system. So the ground beef sample, we try to homogenize before we send it, and you can still see particles there. So there's a multi-level filter. First, it filters all the huge chunks, and then it filters particles above 100 microns. Then it goes to 10. So it's a multi-stage filtration on a small microfluidic platform. Then it comes to that magnetic isolation region where you can pull it with the magnet. You still get a lot of small chunks of things. That's going to mess up your measurement. So it's really the homogeneity of the sample is what determines your capture efficiency in this case. Yeah.
I have a question about the virus that plays such an important role in your bacteria detection. So are people synthesize those viruses or
we get it commercially. We paid $100 to a company. They keep sending us bottles, just like this big of a bottle every year. So it's really cheap. What you can do if you want to synthesize the virus is there is a good reason to synthesize the virus. Our virus is about 100 nanometers tall, maybe 10, 20 nanometers wide, and that is creating. I mean, that is, think of this as a virus, and this as my electrode surface. If one virus particle is going to occupy this much of space, and in case if it's not properly oriented, you actually lose the spatial utilization. But if you really, the component of the virus that attaches to the bacteria is the tail, you don't need the head, right? So if you can just attach, or just make only the tail of the virus and attach to the electrode, you're significantly saving the space, and you can attach more attachment sites on the electrode surface. That way you can increase the capture efficiency. So yes, see, we actually spoke with a molecular biologist who encouraged us to work with them to synthesize just the tail fibers and not the head of the virus. Just by doing genetic engineering, you can not make the whole virus, but only the tail fibers. But when you get something, a kilogram for $100, and you can use it forever, there's really no need to do this synthesis step, which is pretty expensive.
Would it improve the selectivity? I mean, is it possible to tweak your virus?
I'm not sure if it would improve selectivity just because you went from the whole virus to a small fragment of the virus.
I guess I assume there are different types of viruses, and each virus they are more selective.
Okay, so that answers her question is, you don't have to have a single virus. If you want to, let's say I want to simultaneously measure MRSA, Listeria, and E. coli together, I could have all three viruses, a cocktail of virus on the surface. Now, whether you can use the same approach to immobilize all three viruses in terms of the chemistry, that need to be worked out, but it is possible that you could. We have done this for enzymes. We've actually attached multiple enzymes to carry out a cascade of reactions on the surface. Every enzyme does one reaction, and we actually use 3 enzymes to do a series of reactions on the surface to take glucose, convert it into gluconic acid, and then a pyruvate. So we actually went through 3 or 4 steps in that glycolysis pathway on the electrode. So it is possible. You can have, so that way you can have your sensor that is not only selective to one target, but it is selective to a few different targets.
Sorry, I have a very valid question.
We have time. Go ahead.
Threshold for detection, like if the concentration, let's say that the concentration of the virus in a sample is higher than a specific, let's say, the concentration, so the detection can change, or
help me understand.
I'm saying that let's say that you have like an electrode or like a detection technique, So, and then you add a very concentrated sample of virus.
Of virus.
Like a very concentrated sample.
Okay.
Okay, so
when we call sample means the analyte, are you talking about high concentration of bacteria? Or
yes.
Bacteria, okay.
Yeah, so then is there any threshold for this technique to like give error because of the highly concentrated?
Yeah, if you recall, I don't know if I can pull the slides here again. So there is something called a calibration curve here. So here is the linear range. So anywhere from 10 power 2 to 10 power 7, it still falls in the linear range. You can reliably measure. I have not shown about 10 power 7. First of all, it's not needed. 10 power 7, you can begin to sense, smell that's high concentration. But it has sort of plateaus out after this. So it's no longer linear. So we call that as linear range. Oh, you're not seeing that there. Sorry. I don't know how to project it. This one right there. So it's we call it linear range of detection. Within that linear range, you can have a reliable measurement. Outside of that linear range, both at the high end and the low end, it's not as reliable anymore. You can still get a detection, qualitative detection, but it's not going to be precisely quantitative.
Thank you.
Same goes for your glucose meters. Depends on whether you get an AccuCheck or whatever other brands are out there. They do have a specific range in which their measurements are reliable. And outside that range, you can still measure it, but it's not going to be reliable.
All right, then I'll thank our speaker.
Thank you very much.